Cytosections. Researchers, assay developers, and immunohistochemists often have difficulty finding appropriate positive controls for their immunohistochemistry experiments. This problem is exacerbated especially when they are studying new proteins for which a verified tissue sample is not available, or they're developing new immunohistochemistry protocols for proteins expressed transiently or at low levels, or they are screening for new immunohistochemistry antibodies whose staining characteristics is not well established. Lack of appropriate controls often result in inaccurate interpretation of results, leading to high levels of frustration and cost for the project. OriGene offers a solution to this problem. Cytosections. Cytosections are, for researchers, scientists, and immunohistochemistry labs, an ideal alternate control tissue where the expression of target protein is verified. In addition to being a reproducible and renewable source of positive control, it is available for all human and mouse proteins. What are the key features of Cytosections? It is a ready-to-use FFPE section of HEC293T cell pellets overexpressing target proteins. The target protein overexpression is verified by Western blot. It is available for all human and mouse proteins. The target proteins are tagged with MYC-DDK at the C-terminal. All cytosections have appropriate negative controls along with them. Last is it is customizable. Let's get into details of how cytosections are made. OriGene has the largest collection of true open reading frame cDNA clones. These clones are constructed using PCMB6 entry vector that has a MYC-DDK tag at the C-terminal of the protein for EC detection. These clones are transfected into HEC293 cells, cultured for 24 to 48 hours, and the cells are pelleted and verified for target expression using a Western blot. After target validation, these cell pellets are fixed and then embedded in paraffin to prepare FFPE blocks. Thin 4 to 5 micron sections are cut and placed on superfrost slides to generate cytosections. What are the key applications of cytosections? One, it can be used for antibody screening. Two is it can be used when you're developing immunohistochemistry protocols for new protein targets or proteins that are expressed low or at transient levels. In either case, you need a positive control where the expression of the target protein is verified. Cytosection is a convenient, ready-to-use, and renewable source of a positive control where the expression of your target protein is verified. Hence, this is an ideal alternative control tissue that can be easily used for these two applications. Here is an example of a cytosection that is stained for human cytotoxic T lymphocyte associated protein 4 or CTLA4. Figure 1 shows you a cytosection that expresses CTLA4, which is stained for that target using an anti-CTLA4 mouse monoclonal antibody. The dark brown staining around the cell membrane clearly indicates a positive staining. Figure number 2 shows you the same section but stained for the MYC-DDK tag using an anti-DDK mouse monoclonal antibody. Remember that all cytosections have a MYC-DDK tag at the C-terminal of the protein, and that is what is being stained here. The dark brown stain again around the membrane of the cell shows a positive staining. The third figure shows you a cytosection of HEC293 mock transfected cells with vector alone, stained for CTLA4 using an anti-CTLA4 mouse monoclonal antibody. No brown chromogen shows negative staining. In short, this data clearly shows that the target protein, in this case CTLA4, and the MIC-DDK tag can be specifically detected in cytosections, making it a convenient tool for immunohistochemistry experiments. Now, why do customers love a cytosection? First is that it behaves like an FFPE tissue, and it's convenient and ready to use, and hence it's easy to integrate into their routine immunohistochemistry lab workflows. They don't have to do any new standardization of process because it behaves like an FFPE tissue. Second is the target expression is verified. Hence, it becomes an ideal positive control for immunohistochemistry experiments. Third is it's developed from cultured cells in the lab. Hence, it is a renewable source, and it is developed under stringent quality control conditions for consistent performance. Next, it's available for all genome-wide targets and comes with a negative control. The cytosection becomes a one-stop shop for all positive and negative controls for human and mouse proteins. It has a C-terminal of the protein tagged with CMYC-DDK tag, thus offering you an alternate staining target for process control. And custom option is available. That means you can change the tag, or if you want to change the cell pellet composition, or you want to change the cell density to suit your research needs. It's a highly customizable product. Where can you find it? You can find it under proteins under Cytosections. And the catalog number, the SKU starts with TS4, and it has another digits. Then TS4 is also available in packs of 25, so it's called P5. And you can also get the whole block of the Cytosections. And of course, but this is a custom product. It is not available on our website at this point, but you can inquire. In short, Cytosections are a verified, reproducible, and renewable source of positive control. It is available for all human and mouse proteins. It is ready to use 4 to 5 micron thin sections on slides, and it behaves like an FFPE tissue. That is an ideal alternative